Cool. So, hi guys. I'm going to be doing a critical analysis of a paper entitled A Synthetic Oscillatory Network of Transcriptional Regulators. So basically, this is the first study that demonstrated the first synthetic gene network that exhibited stable oscillations. Now, in this study, there's a three transcriptional repressor system. We'll be coming to that a little bit later, but this three transcriptional repressor system, it builds an oscillating network in a culture of E. coli, MC4100. So this transcriptional repressor system, basically it forms this network that induces the synthesis of green fluorescent proteins as a readout of the state of the entire network in each of the individual E. coli cells. So coming to the actual repressors that are mentioned in this system, we have the LACI, TETR, and CI genes. And respectively, they code for the proteins LACI, TETR, and CI. Now, coming to the design of the entire system. So these three transcriptional repressor systems, so you'll have the TETR, LACI, and CI genes. They sort of interact with each other, and this entire system, it's called the repressilator. And it's going to be encoded in a low-copy plasmid. Now, okay, so essentially what happens here is that let's say the LACI gene is initially expressed. So the LACI protein goes and it sort of represses the production of the TETR gene product. The TETR protein represses the production of the CI gene, and the CI gene represses the expression of the LACI gene. So basically, they all interact in sort of like a cyclic manner, and this sort of results in time-dependent oscillations in the concentrations of each of the different proteins. So we have another sort of system in the E. coli. So this is the reporter. So basically, this is encoded by a high-copy plasmid, and what happens here in this sort of plasmid is that we have a GFP gene that's been inserted. Now, to this GFP gene, there's been a TET repressible promoter that's sort of infused to it. So essentially, what this does is that in the presence of the TETR protein, that the TETR protein essentially represses the expression of the GFP. Now, okay, so in terms of the oscillations, which is the readout of the system in itself. So it depends on quite a few factors. There's a dependence on the repressor concentrations. Each of the proteins, the concentration of each of the proteins also affect the action of the network. And these repressor concentrations are dependent on transcription rate as well as translation rates. There's also dependency on the decay rates of the protein and mRNA, and we're going to be coming upon that in subsequent slides as well. So the period of oscillation in these sort of networks is mainly dependent on the protein stability itself. Now, coming to how we're favoring these oscillations and ensuring that these oscillations are happening, there are quite a few, so the oscillations are favored by quite a few factors. So essentially, we want strong promoters and that are coupled to efficient ribosome binding sites. We're also looking for tight transcriptional repression, and we're looking for cooperative repression characteristics and comparable protein and mRNA decay rates. So now we talked about favoring oscillations. Now, to ensure that these oscillations actually happen, there were two things that were modified in the actual E. coli in the plasmids. So we added a lambda PL promoter with LAC and TET operator sequences. This ensured this sort of ensured a tightness in the repressor interaction with the promoter. And we also added a carboxy terminal tag at the end of each repressor gene. Now, adding this carboxy terminal tag, what that did is it sort of reduced the half-life of the protein, the repressors in itself. So, and we mentioned before in favoring oscillations that, we want comparable mRNA and protein decay rates. And so the current mRNA decay rate in E. coli is approximately 2 to 8 minutes, and the addition of this carboxy terminal tag sort of brings it to a comparable standard. Now, so this entire system was, this network was made, it was modeled by transcriptional regulation. It's a simple mathematical model. So basically, a lot of the complex interactions between the molecules, they have not been characterized in this model. But, so in natural gene networks, gene expression networks, there's a lot of noise that's present. And most of the noise is usually associated due to stochastic effects. This sort of, those are like per chance probabilistic events that can occur randomly. So here, so you can see the two graphs that I've mentioned here. So the kinetics of the system was also sort of modeled. And in the graph that I've mentioned here, so in the case without any stochastic simulations, which is the graph above, so you're getting like complete uniform oscillations. But in the event that, once you add any like stochastic simulations to this, you're getting variable amplitudes in the protein concentrations. So in the network design, to ensure that the repressilator is the actual system that's actually producing these oscillations in the protein concentrations, they ran some control experiments. So what they did was they added IPTG in the media, which, as you guys know, it mimics allolactose. And essentially what this does is it affects the repression of the LAC-I protein. And through this, they sort of determined that the oscillations in the protein concentrations happen due to the repressilator itself. Okay, so coming to this, I've made a little truth table, which sort of explained how the oscillations actually happen, why the oscillations actually happen. So here, the binary ones sort of represent the presence of the repressor protein. So let's say in the case that the LAC-I gene is expressed initially. So this is sort of how the system goes. So the LAC-I protein is produced, which in turn inhibits which represses the expression of the TETR protein. And since there's no TETR protein formed in the cell at this point of time, there's, the CI protein gets expressed. Now in the second cycle, since the CI protein has been expressed, it sort of represses the expression of the LAC-I protein. And you can see the cycle sort of like continues similarly like that. And if you look at each column, you're going to have like so for LAC-I, for example, you're going to have a series of 1, 0, 1, 0, 1, 0, and this sort of like continues. And this is how I'm showing you that, oscillations, in terms of concentration, the oscillations actually do occur in this repressilator. Now we'll look at the same case for the reporter, because like we mentioned before, the readout of each state is actually of the GFP. So again, since we had assumed in the previous case that the LAC-I gene is initially expressed, that implied that the TETR protein was not expressed. So considering the TETR protein is not expressed, we're going to have expression of the green fluorescent proteins. And so again, similar to the case mentioned before, there's going to be a cycle of 1, 0, 1, 0, 1, 0, which shows you oscillations in the fluorescence that's sort of produced. Okay, so coming to the actual outputs, the readouts that we're getting. So there are going to be time-dependent oscillations, and these occur with a period of approximately 150 minutes. Now, the important thing to note here is that the state of the network, which implies that the concentrations of each of the proteins and so on, those are transmitted to the progeny cells as well. So, which is sort of why we're seeing an increase in the amplitude for the graph mentioned above. So you can see the oscillations happening, but at the same time, the amplitude of each oscillation is again increasing. This is mainly because the state of the network is transmitted to the progeny cells. Now, the study also found that there were like significant variations in every single cell that they uniquely identified. And so there were a lot of period variations, amplitude variations, and in certain cases there were also phase variations. Now, yeah, so basically there was a lot of noise as well. There was a significant amount of noise. And they concluded that certain changes in the DNA replication can lead to this noise. Okay, so coming to, there's a lot that was really interesting about this paper. So I loved the idea that they sort of modularized biology. They broke it down into like a lot of independent parts, and they built a device out of those parts, and they used that device to sort of mimic. They used the device to make like a little network, and it was a really interesting approach to see so few parts used. And but there were a few issues that I found with the paper. There were a lot of mentions of the words, strong and weak promoters, for example. And there was sort of like a lack of a clear defined standard in terms of measurement. So you're saying strong and weak, but and that's like with respect to something, but we don't really have a measure of what that something really implies. And so, the repressilator was also designed with like a simple model of transcriptional regulation to study its behavior. So a lot of the noise could not be effectively quantified, as well as, you couldn't really get a sense of the molecular dynamics of the entire network. Now, another thing was that, this paper had not varied the host species or the genetic background. So the thing is, because this wasn't really varied, you couldn't really figure out where this sort of network stands within the entire metabolic sort of model itself. So yeah, that's pretty much all I have. Thank you. Okay.
Thank you. Very good. Thank you so much.
Okay.
Okay, so what would be your, what would you do next if you had the chance to continue this project?
Yeah,
How would it would be improved, would be modified?
I would, yeah, definitely like to see, like I mentioned, I'd definitely like to see how the host species and how the entire cell would function if the host species was varied. I'd also want to so the thing is, this wasn't done for like a very large time frame. So there was a mention of, sorry. There was a mention of like how you could have like chemostasis. So sorry, if you like sort of keep the chemical sort of model constant in the entire cell, you would also have like, so the stochastic dynamics would also show some differences. So I would definitely want to, try varying the sort of like maybe like reduce the GFP stability rate as well. Maybe try out a less stable version of GFP as well, because like we'd mentioned before, that you want comparable protein and mRNA decay rates. So
okay, well, thank you. So that's a very good point. So, I think that this paper that as I mentioned yesterday, is one of the foundational papers of synthetic biology, like exposes all the power and at the same time all the downsides of synthetic biology, in the sense that, as your colleague said, the theoretical part can be handled very easily, because you can have a circuit with zeros and ones, and having step 1, step 2, step 3, you make a simulation, everything goes fine. But then at the end, what makes a super big difference is the parameters that you have in the system. So theoretically, you can have a digital setup, but then, whether the protein lasts for a bit longer or for a bit shorter, whether this promoter is stronger, whether the cells are growing fast or are not growing, these are the details that so far are very difficult to tackle. Okay, so that's the bad part of it. So in one of the subsequent talks, I will tell you a little on how, in many cases, parameters cannot be fixed up front, and sometimes one has to use a different trick, that is to enter into the circuit some type of evolutionary approach to let the system to fluctuate until it finds the right parameter set. Okay, so that's something that is quite interesting. And also the other aspect, and I think I mentioned or discussed that with you yesterday, or with one colleague, is that these oscillations last for the period of time that is shown in the paper. But if you let the system to go on, at the end everything will be screwed up, right?
Yeah.
So that's something that will. But do you, in your opinion, do you think that this paper deserves the kind of tag of foundational paper worth a Nobel Prize and everything, or do you think that is so-so?
No, I definitely think I do. It sort of put, the entire idea of synthetic biology, it put in a new light, and it sort of, it tackled those key concepts of deconstructing and reconstructing the system in itself. And I definitely do think it deserves that tag.
Okay, very good. Well, thank you. You did a very good job, and well, thank you so much.
Thank you.
Okay, fantastic. So maybe now you tell us. So you need the computer as well? How do we do it?
Yeah, I have slides. Basically, just put those out in the paper.
Oh yeah, sure. You have a. Okay, fantastic. Very good.
Yeah. So I have only put the diagrams from the paper, and I didn't write much. I thought I'll just explain whatever I understood. So I think this was published at the same time as the repressolator paper in Nature, and was probably the second paper on a synthetically designed circuit using biological components. In this paper, they have reported the design of a genetic toggle switch, which consists of two inducers which act on two promoters. And the two promoters, as in this will have two promoters that mutually repress each other. And then we have a reporter which is a GFP to tell us about the state of the system. Why is it happening like this? So the mathematical model uses ordinary differential equations, and sorry, what is happening? Okay, so this is just we can say the representing the kinetics of the system in the form of Hill equations, but in non-dimensionalized form. So we have alpha 1 and alpha 2 as the okay, so U and V are the concentrations of the repressors, and alpha 1 and alpha 2 are the rates of generation of the repressors, and beta is the cooperativity. So using this model, what they have done is they have plotted the phase space, which means they have equated these two equations to zeros and found out how the concentrations of the two repressors vary. And over here, what they find is equating these two equations to zero, we get three stable points. That is the points where the two curves intersect. And if there are 3 points, also they have analyzed that the 2 points here and here are stable, and the point in between is an unstable steady state, which means when the system is at this point, it will tend to move towards either state 1 or state 2, which they have denoted as the high state and the low state of the switch. So now this kind of a configuration is of course dependent on the value of alpha 1, alpha 2 and beta and gamma. So using appropriate values for these parameters, these 4 parameters, it is possible to get either 3 states, or it is possible to also have values of these parameters for which the system has only one state, which is only one low stable state. So now varying the parameters, they have done a bifurcation analysis, so which means where we look into how the parameter changes affect the movement of these steady states, and they found that it is possible to change the parameters and confine the operation to a bistable region where the system acts like a switch, and then there are some parameter values for which the system will have only one stable state, which refers to this diagram. And this kind of gives us a good idea about what values of alpha 1 and alpha 2 to choose in order to make this system work as a toggle switch. And they have also found how to make sure that this region of bistability is broader and wider, and how it can be reduced into a small region for which the system acts as a toggle switch. So this is the math part of it. Now, to talk about the bio part of it, which I'm not very comfortable with because I don't have much background in bio, but I'll just make an attempt. So this is a plasmid, and they have the initial circuit that I showed implemented here. They have two pairs of repressor and promoter. The first pair they have kept constant as LAC-I and PETRC2, and the second one, the second pair of promoter and repressor, P1 and R1, they have taken as combinations of two types. One is, can you please pass me my notebook? Yeah. So the second pair of promoter and repressor, they have changed to PTAC and PTIC. Sorry, PTAC and pike, and with this they have designed a multiple number of toggle switches, and these to fall into two classes depending on what P1 and PR1 pairs they have taken. So, and this GFP is the reporter. Using this circuit, they have plotted, so first of all, it is possible to induce the toggle action to happen by using two kinds of input, either a temperature pulse, which they have denoted here as 42 degrees centigrade, or it can be a chemical pulse using IPTG. And the gray areas over here denote a pulse duration for this chemical IPTG, and then how the system behaves is it moves towards the high state, and even though the pulse stops here, the system carries on moving towards the upper stable state, upper steady state, which I showed in the previous diagram. And then when there is another input stimulus using the pulse, it goes down to the low state. So in this way, the full toggle action is happening, which is induced by the inputs. And these are different combinations of that for the different, using different pairs of promoter and repressors. And all of them show that they are having the toggle action happening here. So this is some more of the experimental results where they show that it is possible to get bistability, but when the parameters are changed, it is also possible to combine it to a monostable region. But since this has been implemented in a population of cells, then all cells will probably not act simultaneously. So they have given the scatter plot of how the cell population was initially mostly in the high state, and then gradually some of the cells change their state, and then we have a bimodal population, and then eventually over time every cell belongs to the other state, and in this way, like the whole population changes state gradually. So this is again showing over time how the same thing is happening with IPTG as the input, or with temperature pulse as the input. And then here also they show that gradually the population of cell changes, and by 7 hours or so, the full toggle action is complete for the whole population of the cell. So, okay, my slides end over here. So they have said that this kind of a circuit could be applied in practical purposes, probably in a gene therapy setup, but they do not mention exactly how this could be used. So in that sense, I would say that this was pretty basic, and yes, this was because this is the second circuit to be reported, but still we kind of need to find out ways by which we can make use of synthetic circuits like this in practical setups. And since this was in 2000, probably it was not very clear back then how this could be used, but still they do propose that we could probably start building on this. And I think in subsequent years, there have been other papers where they have given more directions on using things like a synthetic circuit for in immune therapy, or so probably yes, we could realize the thing which they projected back then, that it can be usefully made use like applied for practical purposes. So that is all.
Okay, well, thank you. Very good.
Yeah.
So, again, as I asked your colleague, what do you think of the paper? You like it, or it is kind of wishful thinking, or what was your impression when you read the paper?
So I feel that I could mostly relate with the math part of it and not so much of the bio. So I appreciate that in biology they are making use of concepts like bifurcation analysis and all that. So I feel that it really projects that there is a vast area that can benefit from the mathematical analysis that is done in other engineering disciplines. So I totally appreciate that, and I feel good that this is one field which is still not as advanced as physics, so there is room for us to do things and discover stuffs. So yeah, I think the paper does lay the grounds for that.
Okay, well, thank you. Well, thank you so much. And, okay, so well, one thing, please put your presentations in the share. No, you cannot edit that one, right? Okay, so please send me the presentations and I will load them in the shared file. Okay, so everyone has the chance to take a look to them. Okay, so thank you. Well, I want to make a remark about this late work, and it's the following: we are using a toggle switch in one of our projects, where you can see exactly. Well, I don't have a slide for that because I didn't know that you were going to discuss this in the detail that you mentioned. In my laboratory, and I will not give you all the details, we are developing in collaboration with other people a bacterium of Pseudomonas putida. The bacterium called Pseudomonas putida is something that you will hear various times during this course, because I happen to work with this bug, so I'm sorry, but you will hear various times. So this is actually a bacterium that can be manipulated for a large number of industrial and environmental applications. And Pseudomonas putida, in many cases, has been used as an agent for biodegradation of toxic compounds. Okay, so you know that plastics are not toxic by themselves, but they are a big environmental problem. And then plastics are solids, and then for bacteria to degrade them, they have to have access to the surface, and then produce some enzymes that are able, in many cases, to then polymerize the plastics or some of the intermediates produced of its deterioration. Okay, so we have produced a strain in collaboration with a large consortium that is able to degrade PET. PET is this plastic that is used for plastic bottles, okay, in case that you are familiar with it. Okay, so then the question is, how do you make Pseudomonas putida to stick to a surface where it normally doesn't want to stick? So what we made was a genetic circuit that makes the cells to be entirely either in a planktonic state or in a biofilm state. So again, these strange terms. Planktonic means that bacteria are floating freely in a liquid medium. Biofilm is that bacteria are sticking to a solid medium. Okay, so you have a tube, and then the cells that are stuck to the walls are biofilms. The ones that are floating around are planktonic cells. Now, we wanted to control the situation where bacteria stick to the surfaces and the situation where bacteria go planktonic. So what we did was to make a genetic switch, as you have before, in such a way that upon addition of inducer A or inducer B, we can have the whole population sticking to the surface or the whole population leaving the surface. And the trick for doing that is to play with a phenomenon that is known in biofilm biology, and is that the tendency of cells to stick or not to surfaces depends on the intracellular levels of a molecule called cyclic DGMP. So this molecule is regulated by an interplay between a cyclase and a phosphodiesterase. Okay, so what we did was to have, the two kind of expressions that your colleague mentioned, that you can have the switch going in one direction or in the other. In one case, we want to have a cyclase, and the other case, we wanted to have phosphodiesterase. Okay? When you have a lot of cyclase, then the cyclase will produce a lot of the cyclic GMP, and then you have biofilm. When you have a phosphodiesterase, the phosphodiesterase will cleave this compound, and we have a lot of planktonic cells. Well, I'm sorry, my letter is not very clear. But in any case, so by having two genes that either all cells are in a fashion where you have high cyclase, and therefore you have a lot of this compound, then all cells will stick to the surface like crazy. But if you change the inducer, and then you move the whole switch towards the phosphodiesterase, then you destroy this molecule, and then all cells will go into planktonic. So this is something that is important when you think on a process for plastic biodegradation, because at some stage of the project, of the process, you want all cells to really stick to the surface, so the enzymes that degrade the plastics are really in contact with the plastic themselves. But at some point, you want just to release these cells from the surface, and then what you have to do is to use a different thing. One of the tricks that we are entering to this system is that the signals that we want to use for, moving cells in one direction or the other are not chemical inducers, are lights of different colors. Okay, so you have a red light and you have a blue light. So as I mentioned yesterday, there are more and more optogenetic systems that enable expression of given promoters, activation of given promoters in response to lights of different wavelengths. Okay, so the kind of scenario that we are envisioning is one in which, without touching the culture, we can just play with the lights that we illuminate the culture with, and then by playing with the lights, you have the bacteria, either all of them sticking to the plastic or leaving the plastic. Okay, so this is a nice application of this switch, because even though you can say, well, the switch can be applied in the future for gene therapy or for immuno whatever, we are not there. No way immediately, right? So the complications of implementing this type of things in a mammalian system would be immense. If just in this kind of simple system, you have to play with all the parameters and all the things. Imagine when you have a mammalian system or a cell culture, let alone a human patient. So it will be very complicated. At least in industrial microbiology, this is something that is really useful for this type of applications. Okay, so this is one case in which you see that the theory is there, and normally synthetic biologists use as outputs for measurement fluorescent proteins. We all adore, in my laboratory, I don't know whether we have an entire palette of colors: blue, yellow, green, red, you name it. You can make a kind of rainbow with all the colors that are already available. But in the real world, what you want to express is not these nice colors, but activities that make a difference in an industrial setting. So that's something that is really nice and really cool. Okay, so I think that these were two good examples of some foundational papers in the paper. There's still one more paper that I did not mention, and I may bring up to your consideration in a subsequent step. So the three papers were published in year 2000. So the three papers were more or less simultaneous. I have to say that the first paper that was published was yours. Even though I presented first the repressilator, in actual dates, the switch came a bit before the repressilator. There was then another paper in Nature on engineering a feedback control system with genetic tools and so. I may return to that later, or I may even propose this publication as one of the assignments for a future, say, assignment of papers. So that's something very, very good. But you're running out, but doesn't matter, we are over now. Okay, great. Okay, so I think that's basically everything we wanted to discuss for today. I'm still missing your feedback. I have no feedback from you. I don't know what you think of all these things I'm telling you, so I feel like I'm preaching, but I get no feedback. I don't know whether you believe me or you say, well, all these guys saying is bullshit. I don't know, but I would be interested to really know your reaction when I tell you, and maybe for some of you it's not new, but maybe for others it's something that sounds like intriguing, that you can really that have the tools now to think on a serious engineering of biological systems, using the term engineering not as a metaphor, as in genetic engineering. We use the term engineering, but it was not engineering, but as a real methodology. So one thing is to use the term engineering as an analogy versus using engineering as a methodology. And the big change in synthetic biology is that engineering becomes a methodology, not just an analogy. Okay, I think that, well, this may sound like a bit philosophical, but it's really true that because what makes cells to work is a logic relation between the different components of the cell that has to be identical to the logic relation that you have when you have a man-made object, and because it's logical. So there's nothing special or magic about living systems, because it's just the question that the relational logic has to be very similar, even though the materials are very different. So in your opinion, I'm asking you questions, but I never get answers. I'm a bit frustrated. So what do you think is a very, very evident difference between engineered objects and biological objects? I mean, what would be the main difference between this and this? I mean, what's something that will immediately appear? Okay, that would be something, but complexity is a question of numbers, and so I would say that there's not a qualitative difference between this and this in terms of complexity. You are more complex, this is less complex, but still it's the same thing, it's complexity, right?
So from a computational perspective, biological systems are not truly digital, they are more like analog computers. So this presents the perfect stage to have harmonization of analog and digital computing techniques. So I would still say that the logic gates that we are using, they are not very efficient or as efficient as digital computers, because the first thing I noticed was that they take a lot of time to switch. So which digital computers do instantaneously.
Okay, so that's a very good point. So one major difference would be that in this state machine, I mean, you can have a big number of states, but it's still limited because it is digital. Whereas in biological system, you have so many intermediate steps that the number of states that you may have become immense. So that's a good difference. But that's something that you need some insight to make a difference between this gentleman and the computer. I mean, this is something that you, as an expert in computation in biology or in these things, can say. But is there something that to me is far more evident? I don't want to put you under pressure, okay? But I mean, to me, it's something immediately evident. Okay, I'll tell you. That the materials that our colleague has for building his body are soft, and this is hard. Okay? And this may look like an important issue, but this is a super essential matter. Engineers play with screws, with hard materials, with cement, with metals, with things that are hard. Biology work with components that are soft. I mean, there are very few hard things in biology. Maybe the bones, or I don't know, very few things. For most of the cells are soft. We are kind of bland, so, and this makes a tremendous difference in terms of engineering. And you can have, you can have a tree that is very high, but the logic behind this architecture has to be different from the logic that is behind a skyscraper, right? So because the material that you use are different. So the fact that we can abstract some features of biological systems and compare them with features of engineering, may have some limitations because we still don't understand how you can construct complex devices with soft materials. And I think that, in my opinion, and this is more a personal opinion, I think that one of the biggest, there are others that I will tell you in subsequent talks, but to me, one of the major differences between engineering, bona fide, making computers and making planes and everything, and biological engineering is the nature and texture of the materials that are used for construction. Okay, so a cell is about diffusion, is about gluing, is about flexible things. A radio, a computer is completely hard. I mean, you cannot really. However, cells work, and as they work, there must be some logic and some kind of basis for their functioning that I don't think we understand, but it has to be there, right? So I think that one of the challenges for synthetic biology and engineering in the next few years will be how to engineer objects with soft materials, and I think that's something that is super interesting. So so would you agree that your main difference is that you are soft and the computer is hard? You agree? Okay, great. Okay, very good. So the other big difference, and this is something that we can make a list, and maybe at the end I can make a survey of what you think would the main differences between biological engineering and industrial engineering or electrical engineering. One other difference is that in biology everything is context dependent. Okay, so the strength of a promoter, the transcription efficiency, I mean, everything depends on the context, not only the environmental context, that means being a place that is colder or with more solvents or attached to the surface or planktonic. These are environmental contexts, but also you have this context, for instance, in gene expression in terms of chromosomal location. So whether you take the same gene and put it in one place of the chromosome or the other, makes a tremendous difference, okay? So one of the, downsides of this idea of parts in synthetic biology is that by definition, as I mentioned yesterday, part is the minimal biological object that has a defined function that can be associated to a fixed number of parameters or a fixed value for given parameters. But what happens is that it is not completely real because the parameters may change depending on where things are in the cell and depending on many circumstances. And however, it is true also that biological systems are robust. I mean, we are an evidence that we are robust. We can live today and tomorrow and the day after tomorrow, and more or less our circuits, our devices work very well, whereas the capacity that we have to make engineer systems that can be robust is very limited. I mean, you can see it with the repressilator and also with the switch and so on, that over time things will tend not to work well because they are context dependent, they mutate, they are subject to many different variations and so on. So there are some questions there that are interesting. So the kind of standard approach to avoid that is what is called orthogonalization. I will discuss that on Monday. So orthogonalization, this idea that you can isolate completely the devices that we make in the laboratory, put them in the cell, but we engineer them in such a way that they have a minimal interaction with all the signals around them. And by doing that, then the kind of ambition is that these devices will work independently of any other circumstance. It may work in some cases. I don't know whether that's eventually the way to go. Well, anyway, I leave you with these questions. So just to recap and make sure that we understand the program for the next few days. So tomorrow we will not meet, but please remember you have some homework to do. And you may even need to meet in the small groups, either virtually or face-to-face. just to discuss the papers and to decide who's going to make the presentation. And so, please, before we come here on Monday, send me, as as I told you, the list of participants in every group and the name of the one who's going to make the presentation. Please let's do that because it's a very important thing. Okay, so one other thing that I would like to do is eventually to get familiar with your names, because we have to, but to be frank, I don't know what is your name. And I think that's very sad. I think we should, after 2 weeks, we should have a kind of familiarity. We should know each other by the name and so. So let's maybe by you sending me the names and the groups, so I can start identifying who you are and maybe identifying also among yourselves, because I'm not sure that you know entirely each other, right? Okay, I think that's basically all for today. I think I don't know about you. I had a lot of fun yesterday and today discussing synthetic biology, but there's still a lot of more interesting things to come. We're still in what you may call the kind of fundamental part, but the fun will start when we start discussing applications, right? And then you will see the immense power of synthetic biology to solve a large number of issues and also to raise new issues that had never been posed before. One of my favorite issues, and you will have an entire lecture on that, is how to upgrade synthetic biology interventions at a planetary scale. This is one of my main kind of interests in the last few years, and I'm sure that you will be intrigued of some of the things I'm going to tell you at some point. Okay, so that's all for today. Thank you so much for your interest and for sticking around, unlike your colleagues that decided to flee. And I hope that they show up next week at times. And but I think that we should give the assignments only to the ones that are here, because otherwise it might be a bit complicated to get it organized. Okay, have a good weekend, and we'll see you on Monday. Okay.